the cellular tools that generate shape, form, and pattern. Developmental biology in general, and Evo-Devo in particular, are highly interdisciplinary fields that rely on a broad range of specialized research in the biological sciences. For example, since intercellular communication and numerous cellular activities contribute to the generative phenotype and ultimately to the ecological phenotype of each individual, many developmental studies fall under the broad rubric known as cell biology. The development of shape and form, including gastrulation and the formation of the neural tube, as well as critically important migrations of populations of embryonic cells to particular locations, such as the migration of the germline cells to the developing gonads, involve studies in cell motility, changes in shape and form and movements dependent on cellular adhesion and locomotion of cells. All of these aspects of development represent tools that contribute to the generative phenotype, and subtle changes in the regulation of gene expression can lead to dramatic differences in the specific ecological phenotype that results in a particular species. Cells provide a remarkably robust and flexible toolkit, and the functional efficacy of the coordinated multicellular organs produced during development is breathtaking. All of these remarkable results, however, are consistent with and can be explained by the principles of natural selection, EEP, and SSM-IBE that have been discussed in previous chapters. Induction, receptors, and signal cascades. When embryonic cell layers communicate and coordinate their activities to generate unified patterns, they do so by means of a variety of mechanisms, and many aspects of such intercellular communication and signaling are now well understood at a molecular and cellular level. They involve such mechanisms as diffusible proteins, cell surface receptor proteins, and elaborate signaling pathways that ultimately result in coordination of patterns of cell division, motility, and gene expression. The metaphorical tinkering and Rube Goldberg devices that allow such complex pathways to evolve were discussed in Chapter 12. Evo-Devo comes of age, the molecular genetics of fruit fly development as a model system. Ed Lewis takes fruit fly genetics to its pre-molecular Mendelian limit. Building on the stunning success of Thomas Hunt Morgan, one of his students and colleagues, Calvin Bridges discovered the bithorax mutation, which, as the name implies, causes the fruit flies, Drosophila, to develop a duplicated thoracic region. Ed Lewis took Mendelian genetics to the limit. When starting with the bithorax mutation, he discovered and fully characterized what was arguably the most remarkable and historically important complex locus in the animal kingdom, the bithorax complex, BXC. Lewis's pioneering work led to the discovery of the Hox genes that determine the body plans of animals, ranging from invertebrates such as nematode worms and fruit flies to vertebrates ranging from fishes to mammals, including mice and human beings. Hox genes quickly became the quintessential textbook example of developmental toolkits that exhibit deep homology throughout the animal kingdom. Ed Lewis shared the Nobel Prize with Christian Nusslein-Volhard and Eric Wieschaus. With their colleagues, these brilliant pioneers took Lewis's work, which relied on experimental techniques that extended the work of Morgan and colleagues, laid the groundwork for the subsequent molecular genetic studies that ushered in the age of evolutionary developmental biology, Evo-Devo. Nusslein-Volhard and the making of a fly, or a fish, or a mouse, or a human. Evolutionary theory was taken to a whole new level when they used molecular genetics, a powerful combination of techniques from both Mendelian genetics and molecular biology, to perform a systematic screen for embryonic mutations in the fruit fly, Drosophila. These genes had been previously overlooked by other investigators because they cause mutations that are often lethal in early development and are therefore easily missed in genetic screens. Techniques for the in situ visualization of patterns of gene expression. In the biology laboratory, in situ means to examine a phenomenon in the place where it occurs. For example, visualization of a morphogen, a substance whose non-uniform distribution governs the pattern of tissue development during embryogenesis, generally requires in situ imaging techniques that target specific proteins or RNA sequences and make them visible. The development of in situ imaging of morphogens in fruit fly embryos, for example, was a turning point that linked molecular genetics to development and paved the way toward major advances in Evo-Devo. transcription factors, and morphogens. Nussel and Volhard and colleagues applied several powerful new techniques. Briefly, they made it possible for the first time to visualize the expression patterns of several key RNA and protein products, including transcription factors and morphogens. The patterns of expression of these genetic determinants play essential roles in the segmentation and morphogenesis, development of pattern and form of the embryo. This includes the overall formation of the body plan, segmentation, appendages, and local patterning elements of differentiated cells in fruit fly embryos, as well as the bristle patterns and appendages of fruit fly larvae and adults. One of these was already introduced in the previous section on bicoid. Another important example, also discovered by the team headed by Nussel and Volhard, was the discovery of gap genes. Gap genes encode transcription factors that determine segment formation in the embryo by directly controlling the expression of pair-ruled genes.